Hi everyone, welcome back to the channel. And in today's video, we'll be studying about the topic on polysaccharides from biochemistry. So, this is a subtopic under carbohydrates, and we'll be seeing what polysaccharides are. So, polysaccharides, as the name says, poly meaning many, and saccharides meaning sugar molecules. So, many sugar molecules which are linked to one another are called as polysaccharides. So, let's see what the polysaccharide in Greek means. So it means many, and it is made up of many molecules or polymers of monosaccharide units. That is, one sugar units that are linked to each other, they form a polysaccharide. These are usually tasteless, so they do not impart any taste to your tongue, and they form a colloids with water. Colloids in the sense they do not dissolve properly. They form a different form of compound. The polysaccharides can be divided into two types, which are homopolysaccharides and heteropolysaccharides. So the polysaccharide is consisting of a repeating units of monosaccharides, or they have their derivatives, and which are held together by a glycosidic bond. So always remember, there is always a glycosidic bond between two monosaccharides in a carbohydrate molecule or a polysaccharide molecule. Now, the first concern about how what is the function of polysaccharide is, how the structure of the polysaccharide is, and how the storage of energy is. Okay, so these are the two important functions of a polysaccharide, both structurally as well as the storage of the energy. So polysaccharides can be linear as well as branched. They could be a straight chain of monosaccharides, or it could be a branched chain. That is, there can be a branches of the monosaccharide units. So in contrast to the proteins and nucleic acid, these which are usually, these proteins and nucleic acids are usually linear polymers. That is, they are straight polymers. They do not form branches as such compared to polysaccharides. You can see in the diagram, this picture, there is a branching of each chain. So you can see these branches coming out from the main chain. So this is one property or one feature that is seen only in saccharides or polysaccharides. So how does these branches occur in polysaccharides is because of the glycosidic linkage that is formed by the hydroxyl group of monosaccharide. So there is a OH group in a monosaccharide which goes and binds with the other chain of the chain of monosaccharide, in turn forming a glycosidic linkage. So when this glycosidic linkage forms, there is a formation of a branched polysaccharide. Moving to the next classification of polysaccharide, which is homopolysaccharide and heteropolysaccharide. So what is homopolysaccharide? There is a hydrolysis that gives a single type of monosaccharide. So you can see in the diagram, there is a black and white diagram over here, the branched and the unbranched part of homopolysaccharide. So the whole chain will contain a single type of monosaccharide. There is no a different type of monosaccharides being mixed up in the polysaccharides. So these homopolysaccharides are glycans, are polymers of glucose. They could be polymers of glucose, or they could be polymers of fructose, forming fructosan and glucans. So these are the examples of homopolysaccharide. And heteropolysaccharides, as you can see in the diagrams, this is a simpler form of diagram. So you can see there is different types of monosaccharides that are repeated, or they could be non-repetitive. So as in branched and unbranched chain, there are different types of monosaccharides that together hydrolyze and form a glycosidic linkage with each other. Now firstly, the homopolysaccharide, or the single type of monosaccharides that is observed in the chain of polysaccharides. So there are 7 types of homopolysaccharides, which is starch. You can see in the diagram, there is amylose and amylopectin, two forms of starch that together bind to form a bigger molecule of starch. Then we have dextrans, inulin, glycogen, cellulose, and chitin. So these are the 7 different forms of homopolysaccharides that could be given. So focus on starch and glycogen, because most of the time they might ask in your final exams. Moving to the starch. Starch is a carbohydrate reserve of plant, which is most important dietary source for higher animals, including man. So starch is usually a storage unit in the plant, and that starch can be taken as in the form of diet in many higher animals. As in case of herbivorous animals, starch is the main component of their diet. As well as man also consume plants in the form of starch. High content of starch is found in cereals, roots, tubers, and vegetables. So cereals are the different types of the seeds that we consume. Roots, like the carrots, potatoes, tubers, and vegetables as well. So these contain higher amount of starch, and the starch is a homopolymer. As I told, homopolymer will have a single type of monosaccharide. So which single type of monosaccharide is there? We'll see. There is D-glucose unit. So remember, there is D-glucose unit, dextrose rotatory glucose units, and they are held by dextrose glycosidic bonds. So these are the D glucose units along with D glycosidic bonds. And this starch can also be called as glucosan or glucan, because there is monomers of glucose that are linked with a glycosidic bond. Then the starch consists of 2 different types of polysaccharide components, which is water soluble amylose and water insoluble amylopectin. So the 2 different polysaccharide components are amylose and amylopectin. So they might ask the difference between amylose and amylopectin. So please do note, the amylose is water soluble, amylopectin is a water insoluble, and the percentage also 15 to 20% amylose, and amylopectin is 80 to 85%. Then the amylose has an long unbranched chain with 200 to 1000 D glucose units. So amylose has 200 to 1000 D glucose units which are unbranched, and the unbranched in the sense it is a straight chain of glucose units, and which are held by D1 to 4 glycosidic linkage. So the first molecule of carbon is linked with the fourth molecule of another monosaccharide. So that is a linkage, glycosidic linkage between amylose glycosidic bonds. Next, coming to amylopectin, which has D1 to 6. So the first carbon of a monosaccharide or the glucose is linked to the another glucose unit of 6th carbon. So the 1 to 6 linkage is seen in amylopectin, as well as 1 to 4 linkage can also be seen. So amylopectin has both branched and a straight chain of glycosidic bonds. Amylopectin is consisting of a few thousand units, in the branched tree there is around 20 to 30 glucose units per branch. So that is a feature of amylopectin. So how is starch hydrolyzed? So how is starch broken down? So starch can be hydrolyzed by amylase, that is in pancreatic juices and salivary juices. There is presence of amylase which break down and liberate or give out the molecules dextrin and maltose and other glucose, other single monosaccharide glucose units. So starch can be hydrolyzed. Hydrolyzing is to break down the bigger component or the bigger chain of starch into smaller components. So the enzyme that is working on starch is amylase, which are produced either in pancreas or salivary glands, which give out the products dextrin, maltose, glucose units. Whereas amylase is acted upon D1 to 4 glycosidic bonds. So you can see the starch how the linkage is. So there is carbon atom and another carbon atom of two different monosaccharide units which are linked together to form a chain of starch. So there is C1 to 4 linkage and C1 and C6 linkage. C1 and C6 linkage, C1 and C4 linkage in the branched and unbranched chain. So in the unbranched you can see C1 to C4, and in the branched chain you'll see C1 to C6. And coming to dextrins, that this is another form of homopolysaccharide. So when the breakdown of dextrin is taking place, it is released as a product from the starch. So the dextrin is obtained by the action of enzyme amylase over starch, producing a dextrin. Now, starch can be hydrolyzed to produce dextrin, as well as the dextrin can be finally brought into two products as maltose and glucose, further hydrolysis. And the various intermediates that are obtained are usually in the form of soluble starch, amylodextrin, erythrodextrin, and archdextrin. So intermediates are the intermediate compounds that could be formed in the form of in the time of hydrolysis. So this is the molecular structure of dextrin and the amorphous form of dextrin. Now dextrans. Dextrans are polymers of glucose and are usually produced by the microorganisms. So starch is produced from plants, dextrin is produced from starch, and dextrans are produced from microorganisms. So these are used as a plasma volume expanders in transfusion and chromatography. Plasma volume expanders are to increase the volume of the plasma or the cell cytoplasm. So this transfusion, this is used for transfusion in plasma volume expanders, that is to increase the volume of the blood. So dextrans are given as IV through IV for increasing the blood volume. So when there is heavy loss of blood, they could be used as transfusions. And chromatography, chromatography is used in separation of the chromosomes based upon their weight or the length of the DNA. So gel filtration is another example for dextrin usage. Next, coming to inulin. Inulin is a polymer of fructose. So till the dextrans we saw the glucose molecules, polymers of glucose. From here we are going to see the polymers of fructose. That is a fructosan. Fructosan because there is a chain of fructose linked together to form a inulin. So these are produced in dahlia buds. Then you have in garlic and onions. It has a very low molecular weight around 5000 and a polysaccharide which can be soluble in water. Inulin can be utilized by the body and it can be used to assess the kidney function. The GFR rate could be investigated by using the inulin. Moving to glycogen. So glycogen is a storage reserve in the storage part in the animal. So anything that is consumed as glucose is broken down and stored in the human body as glycogen. So that is a storage unit, and it is in high concentration in liver as well as muscles and brain. So it is stored highly in liver, muscles and brain. And glycogen is also found in plants, and they when this glycogen is present in those plants, they do not possess any chlorophyll. They don't have chlorophyll as their component, so such plants will have glycogen. The glycogen structure is similar to amylopectin, which has with more number of branches when compared to the starch. So glucose is the repeating unit of glycogen. So many glucose molecules together form a glycosidic linkage at D1 to 4 glycosidic bond and D1 to 6 glycosidic bond linkages. So these are the two different linkages that is observed between glucose molecule to form glycogen, and the molecular weight is up to 1 micron to 108 microns, and the number of glucose units or the number of glucose molecules that could be seen is up to 25,000 glucose units. The glycogen depends on the source from which the glycogen is obtained. So the glycogen units, the number of units of glucose in the glycogen can be dependent upon from where the glycogen is being obtained. So these are the molecular structures of the glycogen. You can see the molecular structure, the hydrogen and the carbon, oxygen. So you can see here C1 to 4 and C1 to 6 linkage. Now coming to cellulose. So cellulose is exclusively found in plants, that to the plant cell wall. So this is usually observed in the plant kingdom only and not in animal kingdom. Cellulose is not at all present in the animal kingdom. So cellulose is composed of glucose linkage with the linkage of E1 to 4 glycosidic bond, and the cellulose cannot be digested by mammals. Even man cannot digest the cellulose, so consumption of cellulose will just go in waste, but is not completely not digested by the mammals, because they don't have the enzyme that is helpful in cleaving or breaking down the E-glycosidic bond. So that is the reason cellulose is not digested by the mammals, but the certain ruminants and herbivore animals can cleave the E-glycosidic bonds because they produce the enzymes that are involved in breaking of the cellulose. So how is the cellulose hydrolyzed? It is hydrolyzed to produce a disaccharide cellobiose. So it can produce disaccharide cellobiose after hydrolysis. So how is it digested? It is digested by the breaking of the E-glycosidic bond. So the cellulose, though not digested, has great importance in human nutrition, and it is a major constituent of fiber. So the cellulose can act as a roughage. Though it is not digested, it helps in clearing the intestine because it has a fiber content. The intestine is being cleared with the help of this fiber. So this dietary fiber can decrease the absorption of glucose and cholesterol from the intestine, besides increasing the bulk of feces. So it can decrease the more absorption of glucose and cholesterol from the intestine. So it shuts down or decreases the level of absorption of cholesterol or the fats and the glucose molecule from the intestine, and it also increases the amount of feces that is generated. Now coming to the actual, where do you observe the cellulose? So it is usually found in the plant cell wall, around the plant cell wall, and there is a cellulose fiber. Microscopic structure shows some macrofibrils and microfibrils which have the chains of cellulose molecules. So that is the structure of cellulose. Moving to the next type of homopolysaccharide, which is chitin. Chitin is composed of N-acetyl D-glucosamine unit. So till now we saw the glucose and fructose molecules. So this one is another format of polysaccharides consisting of only N-acetyl D-glucosamine units, which are held together by E1 to 4 glycosidic linkage. So this chitin is observed in the shelled organisms or the exoskeleton of invertebrates. Invertebrates are the ones which do not have a vertebra, like insects and crustaceans. So crustaceans are the shelled organism like mollusks. Sometimes it is found in the algae, sometimes in the fungi, sometimes in the nematoda and crustaceans and insects. So this is the chitin that is observed in the different exoskeletons of invertebrates. Now we're moving to the heteropolysaccharides, that is consisting of different types of sugar molecules or the monosaccharides that are linked to each other. So heteropolysaccharides can also be called as heteropolysaccharides or heteroglycans, and the two different forms of heteropolysaccharides, mucopolysaccharides and hyaluronic acid. Now coming mucopolysaccharides, these are the heteroglycans made up of repeating sugar units sugar unit derivatives like amino sugars and uronic acids. So derivative sugars are the ones which are linked with a functional group. So they're not exactly the glucose molecules or the monosaccharides. These monosaccharides can be attached with the derivatives like amino groups and uh, the acidic groups. So they can be attached to the carbon chain, forming a mucopolysaccharide. So these mucopolysaccharide can also be called as glycosamine glycans, glycosamine glycans. So you have to remember mucopolysaccharides also. This is asked frequently in the exams. Then acetylated amino groups. Besides the sulfate and carboxyl group that are present in GAG structure, there is the presence of acetylated amino groups. The acetate is present in the carbon chain. Then the sulfate and carboxyl group together are contributing the acidity of the molecule. There is increased acidic nature because of the sulfate and the carboxyl groups. So COOH and sulfate groups are producing the acidic nature to the mucopolysaccharides. Some of the mucopolysaccharides are found in combination with proteins to form mucoproteins or mucoids or proteoglycans. So these polysaccharides or the mucopolysaccharides can be combined with proteins to produce mucoproteins. So this mucoproteins composition will have 95% of carbohydrates and 5% of proteins. So there is a majority of carbohydrate as well as there is a little amount of protein mixed with the mucopolysaccharide to produce mucoprotein. Now where is this mucopolysaccharide essential? It is a important structure, important component of tissue structure. So it maintains the structure of tissue. So you can see the mucopolysaccharide, which is a glucosaminoglycans. It has a heparin sulfate and dermatan sulfate. So this complex sugar is produced by the cell and is transported to the extracellular space outside the cell, thereby allowing to hold the cells together as well as keeping the tissues together and forming a tissue structure. You can see there is epithelial cell, there is a basement membrane and a connective tissue. So in between the cells and the connective tissue, there is a basement membrane wherein the mucopolysaccharide form a chain-like structure and hold the cells different tissues together. So that is the work of mucopolysaccharide. Now where is this mucopolysaccharide observed? So it is seen in the tissue cartilage, in the skin, in blood vessels and tendons. So these are the different tissue forms where you can observe the mucopolysaccharide. So it consists of collagen, elastin fibers and is embedded in the matrix or the ground substance. Matrix or the ground substance is the part of a cytoplasm that is giving a structure and a proper shape to the tissue or a cell. The ground substance is mostly composed of GAG. The cell's ground substance is mostly composed of mucopolysaccharides or glycosamine glycans. So the mucopolysaccharide is consisting of hyaluronic acid, chondroitin for sulfate, heparin, dermatan sulfate and keratin sulfate are the different forms of mucopolysaccharides. Moving to the hyaluronic acid. So hyaluronic acid is an important GAG and it is found in the ground substance of a synovial fluid of joints and vitreous humor of eyes. So it is found in the joints. So you have heard of synovial fluids in between the joints. So these keeps the joints in a perfect motion and they do not cause rubbing of the two bones. So this fluid keeps the joints moist and like an oil for an engine to work efficiently. These also act efficiently. And vitreous humor of eye is important for the eye's nutritive supply as well as keeping the eyes fresh. So that is how hyaluronic acid helps. It is also present as a ground substance in the connective tissue and forms a gel around the ovum. So it is found as a ground substance in connective tissue. The tissue between the epithelial cells and connective tissue has a layer of this hyaluronic acid which forms a ground substance, and it also forms as a gel around the ovum. So it is forming a gel around the ovum to protect the ovum, and it keeps the ovum fresh and healthy. Hyaluronic acid, as I said, it acts as a lubricant and shock absorbent in joints. So it helps in smooth functioning of the joints and also acts as a shock absorbent whenever there is a sudden touch or a sudden a jerk on your joints. There is this hyaluronic acid which is absorbing these painful accidents that happen near the joints. So that is a work of hyaluronic acid. Now you can see the hyaluronic acid, how it is observed in from a naked eye, and how the chemical structure that is observed. So this is the hyaluronic acid. So this is the end of the topic on polysaccharides, and hope you understood the video. If you didn't understand, just go through the video once again, and I have made the notes for each and every topic which could be observed in your final exam. Kindly note every topic into your notes and keep studying for the exam. Thank you.